Welcome to Click for IB Biology. We're studying topic C3.2, Defense Against Disease. And these are actually infectious diseases. And in this topic, we're going to be taking a look at the details of the immune system and its functioning. There's a PDF of this presentation that's linked into the description, which is below the video. And there you can also find 10 questions, a quiz, short answer questions that will allow you to secure your understanding. The answers are also provided. So let's take a look at the immune system and the challenges to the immune system. And those challenges take the form of the disease-causing organisms, which are technically known as pathogens. I have 6 groups of pathogens in my diagram. The prions are not discussed on this course any further, but they are known to bring about malfunctions in protein folding, such as in Jacobs-Creutzfeldt's disease. However, we won't consider that one any further. We do also have, for higher level students, there's the topic A2.3. So this is a higher level topic only about viruses. We will be looking in this study, in this particular topic, at viruses as well, but higher level students should cross-reference to that particular topic. So we'll look at those ones. Prokaryotic diseases are those of the bacteria, here illustrated by Vibrio cholerae, of course the causal agent of the disease, cholera. Our fourth group are the fungi, here illustrated by Trichophyton. Trichophyton is responsible for athlete's foot, whereas Candida albicans is more associated with yeast infections and typically then of the reproductive system. Single-celled eukaryotic cells are the protists, and our example here given is Plasmodium, which is the causal agent of the disease malaria. Let's move to something a bit more elaborate and multicellular. We have the nematode worms in the forms of Ascaris, perhaps more typically known as infecting the digestive systems of dogs, but Ascaris, of course, does affect humans as well. Tinea is the tapeworm, a much more complex organism, which could be seen as an example of the helminth worm pathogens. Now, these pathogens come up against our immune system. Our immune system is organized into barriers to infection. So the first attempt is not to let those pathogens enter into our biological system at all. And skin is a great barrier to that process. So the dryness of skin, the dead layers of skin, but also the oils secreted from the sebaceous gland discourage pathogens by repelling water, of course. Mucus, which is in our mouth and our tracheal system, traps microorganisms as well. You'll be familiar with mucus in the lining of the trachea of the respiratory system. But of course, you'll remember that on swallowing, any trapped microorganisms go down into our stomach, where they would actually encounter hydrochloric acid. So that's another example of a barrier to infection, but that's where we're going to leave this particular feature and concentrate on the 3 other items under the immune system, looking first in this video at clotting and what we can do if the microorganism, if the pathogen manages to get past the barriers to infection. So if the pathogen gets past those barriers, is there a response that we can produce? So we'll look at that in this particular video. In the next video, we will consider the innate immune system. Now, this is a rapid response to a pathogen gaining entry into the body. It's non-specific, so it's a generalized system dominated by the process of phagocytosis, in which white blood cells engulf pathogens. So that will be a video for us to look at as well. And then the adaptive system, the adaptive immune system, is confined to those organisms which are the vertebrates. And we find that where there is a humoral response of B cells producing antibodies and a cytotoxic response of T cells producing cytotoxic T cells to fight tissues infected with viruses. Both of those systems provide us with immunological memory, and these three items are also dealt with in later videos. So let's take a look at the clotting process. So when we have a wound, then wound tissue sends out chemical signals, or alternatively, we have damage to the inside of a blood vessel, and that equally will send out chemical signals, and these attract the platelets, which is one of the parts of the blood system. Now, the platelets are not typically cellular, they're quite small, and they have no nucleus, cell membranes intact, the organelles are greatly reduced. But what the platelet can do is produce what are called clotting factors. So what's going to happen is that in response to the wounding signals or the damage to a blood vessel, the platelets release a clotting factor, and we're now going to enter into what is called a cascade of events. Now, for the purposes of simplicity, we've reduced it to the ones present in the diagram, but there are actually multiple steps. And the platelet converts plasma prothrombin, which is in an inactivated enzyme, converts this to the active enzyme form called thrombin. And in turn, as part of our cascade, thrombin turns the soluble plasma protein fibrinogen, is converted to an insoluble protein, fibrous protein called fibrin, appropriately. Now, fibrin creates a meshwork of protein molecules like so in the wound structure, and then the consequence is that red blood cells, drawn clumsily here, become trapped within this fiber, and this solidifies, and that's what we call a clot. So this can happen in the wound, and it can also happen in a damaged blood vessel, and that gives us our second tier response when a pathogen has managed to breach the barriers to infection. And so in our next video, we'll take a look at how any pathogens replicating within our tissues or within our bloodstream are dealt with by the innate immune system through phagocytosis. The link is there. Link to subscribe to the channel, please, greatly appreciated. And the playlist is linked on the right-hand side for your convenience.